Okay, eukaryotic gene regulation. In our outline, we'll discuss levels of it, and spatial gene regulation in plants, temporal gene regulation in animals, chromatin structure, how enhancers regulate genes, transcription factors in gene regulation, then messenger RNA splicing, messenger RNA stability, and lastly, RNA interference. Okay, eukaryotic genes can be regulated at several levels: the structure of the chromatin, transcription, of course, what's done to the messenger RNA, and lastly, post-translational modifications, what's done to the protein after it's been made. We're not really going to talk about that, though. It's beyond the scope of our discussion here. So, stages of gene regulation shown here. We can do it with regards to transcription and RNA processing inside the cell nucleus, and then with regards to translation and post-translational modifications out in the cytoplasm. So spatial gene regulation in plants. Tubulin genes are found in the plant Arabidopsis thaliana. This is a plant that's been studied a lot by geneticists. It's kind of the fruit fly of the plant world. Dr. Wrench works with it quite a bit. But anyway, tubulins are proteins that are a dimer consisting of one alpha tubulin and one beta tubulin from different sets of genes. And these tubulins are dimers forming long chains that make up microtubules that are used a lot in different cellular processes. There are 6 alpha tubulin genes in Arabidopsis and 9 beta tubulin genes, and there are different genes are expressed in different cells in the plant's body. So for instance, a gene called TUA1 is an alpha tubulin gene. It's expressed in pollen grains and young anthers, but there's little or no expression of it in the leaves, stems, and roots of the plant. TUB1, a beta tubulin gene, is expressed in the roots just above the rapidly dividing root tip, and TUB8 is expressed in the vascular tissue, the xylem and phloem. And this shows the expression of TUA1 in the anthers, the male reproductive organs of the flower. You can see the rest of the flower is just unstained and white, but the anthers are dark, indicating gene expression there in that part of the plant. So, temporal gene regulation in animals, then moving on to that. We'll discuss the globin gene family, but first we need to define what gene families are. These are sets of closely related genes with slightly different functions, and they arose from a single ancestor via accession of gene duplication events in the past. Hemoglobin and myoglobin make up the globin gene family, and there are embryonic hemoglobins, fetal hemoglobins, and adult hemoglobins. So these genes are turned on during different parts of animals' life cycle. And there are, of course, different alpha and beta genes for each on different chromosomes even. This shows the human globin genes. So on the top row, the alpha cluster is on chromosome 16. The beta cluster is on chromosome 11 in the bottom row. Embryonic hemoglobins are expressed earliest. They're shown in yellow there, followed by fetal hemoglobins are expressed next. Adult hemoglobins in red. Then that theta hemoglobin in chromosome 16, we don't know what it does. And the black ones are pseudogenes. These pseudogenes are DNA sequences that have gene-like elements in them, like start and stop codons, promoters, etc., but they don't seem to be functioning at all. So they may have been genes in the past that have mutated to become non-functional. Anyway, we call those pseudogenes. So this shows the transcription units. In other words, what the RNAs look like in various different genes. And you can see that first, the exons are in green here, the introns in white, and the UTRs are in that tan color. But you can see that the first exons and first introns are very similar in size in these various genes as the second introns. The second exon is as well, and even that last exon they're showing you in green there are all similar in size. So this shows you that the RNAs that these genes make are similar but not identical, because the genes themselves are similar but not identical. So it shows an evolutionary history of the globin gene family. They were all clustered together to about 500 million years ago when the alpha genes moved via transposition. We'll talk about what that is later on, from chromosome 11 to 16, and the beta chain remained on chromosome 11. And then we can see divergence about 200 million years ago between the fetal and adult hemoglobins of the beta part of the family. And this shows you the timing of gene expression. So the alpha cluster in red lines, beta cluster in blue lines. You can see the vertical line in the middle there is birth, and so to the left of that is post-conceptual age in weeks, and to the right is postnatal age, also in weeks. So some of these hemoglobins start and stop their gene expression right at conceptions, and others wait till much longer in time. So different genes are expressed at different parts of the early life cycle in animals. That's what this example is illustrating. And so here's our final hemoglobin molecule. Remember, a tetrameric molecule with 2 beta chains and 2 alpha chains. So with regards to the chromatin structure, chromatin remodeling refers to nucleosomes being moved, or the histone portions of them being moved, exposing different parts of the DNA, and this can expose promoter regions and so forth to transcription factors and RNA polymerases. And it says there's been studied best in yeast and fruit flies and in humans. So this kind of shows you that this remodeling complex is a proteinaceous complex that requires ATP, and we can see it's sort of like moving the nucleosomes out of the way so that the RNA polymerases and transcription factors then can bind where they need to bind to. This little yellow segment here, for instance, is the promoter region of a certain gene. We can also chemically modify the histones in the nucleosomes, particularly their tails. And this can either suppress or activate transcription. So things like phosphate groups, methyl groups, acetyl groups, or ubiquitin can be added or removed from these histone tails, and that will alter their chemistry somewhat. So we can see here, so the tails are these little things that stick up, and in number 2 there, number 1, positively charged tails are, of course, are attracted to the negatively charged DNA, and they want to wrap around it. But we can free those things up with acetylation, making those histone tails less positive, and then they won't have such a strong attraction to the DNA, thereby freeing up some of that DNA so that transcription factors and RNA polymerases can bind to it. Moving on to enhancers then. These are DNA sequences located kind of near the gene being regulated, and proteins called special transcription factors bind to them. So basal transcription factors bind to the core promoter region, or what's often called the core transcription apparatus that consists of the RNA polymerase as well as these transcription factors that bind right next to the gene. So these enhancers can act over a relatively large distance, up to 50,000 base pairs away from the gene. They can be inverted, flipped over, and they still work. So they're fairly flexible. And their effects are even independent of their position. They can be upstream from the gene, downstream from a gene. They can even be within an intron, and they still function normally, apparently. So, but a lot of them function in a tissue-specific way. They will stimulate transcription only in certain tissues. So, for instance, in fruit flies, they have a yellow gene that is recessive. When expressed, it makes the wings, legs, so abdomen and thorax, makes the fly yellow. And where wild-type mutants are more of a tan to brown color. And then there are mosaics that have some yellow on them and some not. And this is due to enhancers being mutated in certain tissues, but not others. And so the transcription of this gene occurs only in some of the fly's body and not in others. This shows you the first couple exons of the Drosophila yellow gene. And the enhancers you can see are upstream there. There's one for wings, another for thorax and abdomen, another for the fly's larval body coloration. And then intron 1 turns out to be an enhancer for bristles, for tarsal claws, and for aristae on the fly's body. So in this case, the intron is an enhancer. Now, associated with enhancers are these sequences called insulators for an insulator binding protein. And so this shows you that enhancer 1 only can regulate gene A and it's blocked from regulating gene B. Enhancer 2 can only regulate gene B and can be blocked from regulating gene A. So these enhancers can be not only tissue specific in their regulation effects, but also gene specific in their regulation effects. And here's a yellow fly on the bottom and then a wild type fruit fly on top, showing you it's got some brown and black on it. I couldn't find a picture of a mosaic yellow fly to show you, however. So transcription factors, as we know, are proteins important in transcription, and some of them have homeodomains. These are conserved regions of 60 amino acids, so that'd be 180 base pairs, that have a helix turn helix motif. Remember, we talked about that before in chapter 16. And mutation in these homeodomain genes alter the developmental fates of cell groups. So for instance, mutations in the antennapedia gene cause the antennae to become legs. Cool. And so it activates the gene, but it activates it in the wrong place, in the wrong part of the fly's body. And so here's our core transcription, our transcription apparatus, showing you the core basal apparatus down there. At the bottom, the long orangey hot dog thing is the RNA polymerase molecule, and then all those other things are transcription factors, all the little circles and ovals of different colors. And so here's that helix turn helix motif. Remember, that's how the transcription factor physically binds to the DNA, as you can see in this lower part of this diagram. And so here's the head of the mutated fruit fly on the right and the wild type on the left. So the head is normal. It's just that it's grown a normal pair of middle legs, but it's grown them out of its head where the antenna should be growing. So when you mess with those homeodomains, you mess with the messaging system in the fly that tells it where to make the stuff. It makes the right stuff, it just makes it in the wrong place. So moving on to alternate RNA splicing then. Certain exons can be left out of an RNA transcript and you can get a slightly different protein from the same gene that way. There can also be alternate 3' cleavage sites in the messenger RNA molecule too, which can affect the protein structure. So this is regulation at the level of RNA processing, past transcription, but before translation. So one example comes from the rat T troponin gene, and it's a protein found in the muscle of rats. And the finished protein can be anywhere from 150 to 250 amino acid, depending on which exons are included in the final messenger RNA and which are left out. And that's what this shows. So there are 18 exons in this rat troponin T gene. All the blue ones are always there, but various combinations of the yellow and green ones are present in different functional mature messenger RNA. So you can see the first one has exons 4 and 5 in it, and 7 and 16. The second one's got 4, 6, and 8, and 16. And the last one's got 4, 6, and 8, and exon 17 in there, giving you different proteins from the same gene and messenger RNA molecule for that matter. This shows you how we've got 2 3' cleavage sites. So this gene's got 6 exons as we see on top. There's its pre-messenger RNA, all the exons are there, but there's a 3' cleavage site at the 3' end of exon 4 and another one at the 3' side of exon 6. And in thyroid cells, the first 4 exons are combined together to form the protein calcitonin. But in brain cells, exons 1 through 3 and 5 and 6 are combined to form calcitonin gene-related peptide, the CGRP, which, again, both come from the same gene, and the RNAs are just processed differently in different cells, yielding different proteins. And here we have something similar, showing you 2 3' cleavage sites, both in exon 2, let's see, cleavage site 1 is upstream from cleavage site 2. And so you're going to get a shorter mRNA from cleavage site 1 than you are from cleavage site 2, and therefore a different, a slightly different protein in the end product there, because the mRNAs are the one in the number 2 there is longer than the one in number 1, of course. Okay. Now we can look at RNA stability. So this refers to how long the messenger RNA molecule lasts in the cytoplasm. So obviously long-lived messenger RNAs will support much more translation polypeptide synthesis, and short-lived ones will not. So we're controlling the level of gene expression by controlling the rate of messenger RNA degradation. How long does that molecule, that is the mRNA, last in the cell cytoplasm before being degraded? So there are several factors that can determine messenger RNA longevity. One seems to be the length of the poly A tail. It's very, very short in histone proteins, or they lack them. And so these histone mRNAs are very short-lived. The structure of the 3' UTR can also affect the stability of the messenger RNAs, and then the metabolic state of the cell. So for instance, in female frogs, estrogen increases the longevity of vitellogenin gene messenger RNA. And now vitellogenin is an important protein in the yolk of eggs. And so as the breeding season is approaching, the female frog is going to be needing to yolk up her eggs. And so an increase in estrogen in her cells will increase the amount of vitellogenin protein that then she can put into the eggs, the yolk of her eggs. So that's RNA stability. And lastly, RNA interference. There's an enzyme called dicer, and it can make mi and siRNAs. Those stand for microRNAs and small interfering RNAs, respectively. They're small RNAs, 25 base pairs or less. They combine with proteins to form a RNA-induced silencing complex, or RISC. And then this RISC can cleave the messenger RNA, causing its degradation, or it can bind to the mRNA, causing or interfering with transcription or translation. And this shows RNA silencing by cleavage. So there's our dicer enzyme up there, and it helps form RISC by chopping up this RNA into small interfering RNAs, and then those can combine with the RNA molecule and start to cut it, cleave it, and then it will be degraded, and there will be no more translation occurring from this molecule. There can be translation inhibition as well, where the microRNAs can combine with the RISC and then pair with the messenger RNA to lead to an inhibition of translation. And then lastly, a similar compound, RNA-inducing or interfering transcription enzyme RITS, can combine with the siRNA to methylate the DNA, and that can inhibit transcription, as we see the methyl group shown as a little ball and sticks down at the bottom. And so that's all I have on eukaryotic gene regulation.